All right, thank you, Mark. We'll give you a little bit of a well-deserved break. So I think we can all agree this morning's session was outstanding, and we have another 3 outstanding talks coming up. The first is going to be Nick Loman from the University of Birmingham, who's going to talk to us about real-time microbial source attribution with sequencing in space.
Thanks very much, Jen. Thanks, Mark. Thanks to the whole NASA crew for getting us here, and also to Nature for having us along. So I'm going to talk about source attribution with sequencing and strain tracking, and try and refer this to space if I can. I'm not a massive space nerd, but I think it's so exciting when you come here and you look around and get really excited about the prospects of deep space travel. Although I don't really fancy it myself in that little Orion thing, whatever. Okay. I'm at the University of Birmingham, that's in the UK, not Alabama, just to be clear, in case you couldn't tell from my accent. And I'm also a fellow at the Alan Turing Institute. Okay, so this is like the slide I like to use in all my talks, just because it kind of settles me. This is the big idea, which is in the future, in clinical microbiology, in outbreak surveillance, we're going to just go for sequencing first. So we're going to get rid of culture. We're not going to waste our time with rapid diagnostic tests. We're not going to go and do metabolomics or proteomics. We're just going to do sequencing in clinical microbiology when a patient presents and we are concerned that they have an infectious cause, or maybe even a non-infectious cause. And Charles will talk about this. And the reason I think sequencing is going to be an important test in clinical microbiology as a first line is because only sequencing and through generation of genomes and comparing genomes gives us so much rich information that is useful for a clinician or useful for an epidemiologist, and in fact useful for biologists and people that study evolution. And I've kind of separated into these 5 separate categories of information that we can get. And I'm not going to talk about diagnosis at all because Charles is going to talk about that later on. I just want to mention phenotypic prediction. Phenotypic prediction from genomes is difficult, will continue to be difficult, but for important clinical phenotypes like antibiotic resistance, for example, for some organisms like TB and Staph aureus, we're actually quite good. We're actually as good as in vitro methods at determining phenotypes just from genome data. And if we add in things like gene expression data, we can get even better. So that's an important use of getting genomes. And obviously, in the example of TB, we can sequence a TB genome much more quickly than we can isolate it and do antibiotic phenotypic testing on plates. The subject of this talk, though, is going to be mainly about the idea of using genomes for epidemiology, and we can exploit the fact that microorganisms evolve at reasonably fast rates, anything from a few SNPs a week to much slower rates, but we can use that generation of genetic diversity to allow us to draw phylogenies and to do clustering and to see how cases relate to each other, and critically to do source attribution. Where does something come from? And that's obviously important in outbreak and epidemic response. So I'm going to mainly focus on that and the more fine-grained data we can get in terms of how cases link to each other. And then finally, I'm not really going to talk about this, but it's an important thing. How do our interventions, human interventions in terms of treatment, vaccinations, affect microbial populations and selection pressure we put under, and what's the link with host genetic susceptibility and host response? Okay, so next slide, please. I'll go on. Is this my laptop's fault? Okay, all right. So a bit of space. I say I'm not a space nerd, but it is really cool to be here. So this is a picture from the Apollo 10 crew that were training in a command module simulator. NASA people, who was on the Apollo 10 crew, please? Oh, they're all dead. Oh, dead. Yeah, okay, but who were they? Yeah. No, what are the names of the people on Apollo 10? Shout out. NASA nerds, I'm disappointed. No, you don't look it up. You're right. Okay, I'm just a bit disappointed. I know because I prepared these slides in advance. But I didn't want to like come and tell NASA who was on the Apollo 10 mission because it just seems wrong, right? Yeah. So okay. So Stafford, Young, and Cernan were on this mission. Okay. And and I'm really pleased that no one else has found this story already because I was sure it was going to come up already. But on this mission, which was a success, 6 days in, the astronauts were in lunar orbit, and something rather perturbing happened. Does anyone know this story? Okay. Well, I've got what happened is basically, and this is recorded. Tom Stafford said, "Oh, who did it?" "Who did what?" asked John Young. "Where did that come from?" interjected lunar module pilot Eugene Cernan. "Give me a napkin, quick," commanded Stafford. "There's a turd floating through the air." Okay, next slide, please. And then there's these transcripts that that you can get. Yeah, so I didn't do it. It's not one of mine. I don't think it's one of mine. Mine was a little more sticky than that. I love that. Throw that away. Was there? There's a video too. Well, there you go. This is from an article I found on Vox.com. Next slide, please. This carries on, actually, this conversation about the turd. And, and I guess the point about this is that is source attribution important in space? Well, it was very important to these astronauts, at least. So if they'd had the ability to sequence these turds and relate it to a database, perhaps they could have worked out for sure who was responsible. Okay, I've looked at this slide. I've looked at this picture in a lot of detail, actually. And this is from Apollo 11, obviously, not Apollo 10. Oh, my presentation's gone terribly wrong, hasn't it? I'm just going to do stand up for the rest of them. Can you do? Is it your bit, or is it my bit? Do you think? Oh, you're good. Okay, so that's fine. And can you just advance one slide? I've looked at this slide in a bit of detail, and I think that there's something dubious about this picture. I think they missed something. All right, that was a poo. Right, if you didn't see it. Okay, so serious stuff now, slightly more serious stuff. I want to just try and sell to you, I guess what I'm selling today is the idea of source attribution and epidemiology and strain tracking. And I'm going to give you two hopefully quick case studies. And of course, it relates somewhat to what Julie already beautifully presented. But hopefully it will strengthen that idea that genomic epidemiology is a really important tool. Yeah, next slide, please. Okay, so we're firstly going to talk about Pseudomonas aeruginosa, and there's another space link with Pseudomonas aeruginosa that Cheryl reminded me of, which is that Fred Hayes on Apollo 13 had a Pseudomonas urinary tract infection, so thank you for that note. I didn't remember that. But Pseudomonas is a big problem in the hospital environment, and of course Pseudomonas is very commonly associated with populations in water, and although it predominantly affects patients that are hospitalized, that are sick, immunocompromised, or particularly with burn injuries, although I suppose burn injuries are possible in space, this is an important MDR organism that we need to worry about. And the importance of Pseudomonas in the UK really came to prominence associated with an outbreak in Northern Ireland that affected newborn babies on a neonatal ICU. And as is so common with Pseudomonas, which has been happening since, at least since antibiotics have been used in clinical practice, since the sort of 50s and 60s, Pseudomonas was linked to the water supply. And when the investigators dug into the water supply, they found what we see so often, which is biofilms, and biofilms affecting plumbing parts, like, for example, flow straighteners and shower heads. And they're absolutely teeming with Pseudomonas. And of course, a patient that is susceptible to an infection can develop an infection from planktonic cells coming off that biofilm and infected them. Next slide, please. So, Pseudomonas, just to sort of re-emphasize that point that Julie made, including, is one of a number of important organisms that live in biofilms, can grow co-associated with amoebae, grow as oligotrophs, and grow under conditions of stagnation. And Legionella is another one that's been mentioned at this meeting, and Acinetobacter and as Pseudomonas, as Julie mentioned. So these are important putative opportunistic pathogens, and they're associated with water. Next slide. And so we wanted to look at, so Pseudomonas is actually particularly a problem in our hospital in Birmingham, and we've been tracking Pseudomonas infections, of which we have a high rate in Birmingham, for quite some time. And we wanted to really try and understand why we had so much Pseudomonas in Birmingham. And so the epidemiology here is that that we have a new hospital at the top here, that was built in 2010. It's got actually the largest ITU in Europe. And when we sample assets, we sample taps from Birmingham ICU, we find a very, very high positivity rate. And interestingly, if we compare to an older hospital built in 1975, this hospital, which has various differences, including a copper treatment system for the water, has a very low Pseudomonas rate. And this seems to correlate epidemiologically with the number of Pseudomonas infections we have as well. So this study, we looked at 4 hospitals. These other 2 hospitals have kind of intermediate rates. Next slide. If we look at the whole genome analysis and just divide these into kind of rough clades, which are color-coded here for your convenience, we see quite diverse sequence types and clusters across this tree, but we also see certain clones, particularly the one in red, which is ST179, almost entirely associated with Hospital W, our hospital in Birmingham. You can see other evidence of kind of clonal expansions associated with particular locations. Next slide. And if we draw this onto a map of our 4 ITUs, what we have here is we've got 4 ITUs that are arranged contiguously. And what we've done here is we've sampled water from the hospitals at 3 time points over the period of 3 months. And the simple thing here is just to check, if we sample the same water outlet over 3 time points, do you see the same clone in the same tap or in the same water-associated environment? And we do. Effectively, taps seem to be quite commonly persistently colonized, and you see the same clone in the same place at different sampling periods. Next time point. Next slide, sorry. This is just a zoomed-in version to show you that that's the case. You see these different lineages associated with different taps. Next slide. Now, if we look at kind of like genetic distance here, what we're doing here is we're comparing between all pairs of isolates between hospitals and within hospitals, between wards and within wards, and between outlets and within outlets. And what we can see is actually wards don't look, within a hospital don't actually look particularly similar. They're not that much dissimilar than between hospitals. You can obviously see, because of those big, kind of clones that within the same hospital, we do see more genetic similarity. But the striking thing here is that if you look on a per outlet basis, you see very, very little genetic difference between water samples that you collect at different time points. So next slide. That's within the same hospital, yeah. Yeah. And so what that's showing is that each outlet potentially has a unique signature, a unique population of Pseudomonas. And if we look right down into fine-grained kind of whole genome sequence phylogenies, where we, like Julie did, try and eke out as many of the possible SNPs we can in the genome, we can draw these very fine resolution trees. And you can see that within these fine resolution trees, these color coding on the right here is clustered by bed space. You can see we have clusters, we have individual subclones or individual sublineages within that larger lineage that associate with particular bed spaces. And in some cases, we can see examples of where you have a patient infection here in red in this first column, where you have this indistinguishable isolates from the patient as to the ones found in the water supply, in this case, in a shower unit and the environment around the shower. And in fact, if we go back to the records, we can see that this patient that was in this bed space 11, this patient was in bed space 11, and their cases cluster with the water isolates from bed space 11. So these are only a few, these are small numbers of SNPs, you know, maybe like 20 or 30 SNPs to build this tree in its entirety. Next slide. And if we look into this a bit more detail, what we see is that the early isolates from a patient are actually indistinguishable from the isolates that we see, that we can collect from the shower and collect from the environment around the shower. But quite quickly, within a patient's infection, we see signatures of adaptation associated with treatment of antibiotics. So in this case, the patient was giving meropenem, and we can see actually there's only about three or four SNPs total detectable from this patient over the course of treatment, but those are important mutations. For example, deletion, single-base deletion in OPRD, which is an outer membrane porin, very important for imipenem, meropenem resistance. So we see a small number of SNPs, and these are very clearly adaptive changes. Sorry, so red means from a patient, blue means from the water, and green means from the hospital environment, the water environment, like the around the patient. Yeah. Sorry, I didn't make it clear. Okay, next slide. Okay, so how long have I been talking for? About 2 hours already. No, I won't have time to talk about. So basically, short source attribution works really, really well. It works well for these hospital outbreaks. It also works really well for foodborne pathogens. So in the US and the UK, all foodborne pathogens, E. coli, Salmonella, Listeria, are getting routinely sequenced and used for doing cluster identification to identify contaminated food sources. In Birmingham, we're also using whole genome sequencing routinely for Mycobacterium tuberculosis clustering and AMR prediction. And our vision, I guess, is that we'll have sequencing available in real time to help with epidemiology, detection of outbreaks, and outbreak response. So I'm going to quickly talk about Ebola virus in terms of outbreak response. And just the power of using the sequencing data to try and look at fine-grained transmission within an outbreak. Okay, so this slide is basically an epidemic curve at the bottom showing cases in thousands, and at the top showing that during the Ebola virus epidemic 2013-2016, there were really very few genome sequences available. Next slide. Does this work now? Oh, it does work now, brilliant. Okay, our solution to this problem of how do we get genome data in the Ebola virus epidemic is to try and deploy sequencing close to the sample. So rather than trying to expatriate samples from an outbreak area using quite reluctant courier services like FedEx, instead we would take the sequencer to the sample. And of course, this is analogous to what NASA want to do and have been doing with the Oxford Nanopore MinION, which is what we used as well. And we found that actually a very small amount of equipment is required to do sequencing in a field-based situation, working with already made safe diagnostic samples. So this is all you really need to get a basic sequencing lab set up. And this is just my PhD students' pants and socks. You don't need that for DNA sequencing. It's useful to have a change of clothes. Okay, so quickly, this epidemic raged from 2014 through 2015 and finished finally in 2016. And so at the start of 2015, we were able to establish, working with Public Health England, working with Bernard Knox Institute, a sequencing laboratory, actually, in fact, various laboratories. And so Josh here went to set this up, working with Miles Carroll from PHE. And working with Miles Carroll was Sophie Durifor, who absorbed this technology and set up a real-time genome sequencing surveillance setup, training local Guinean scientists, Joseph and Raymond, and volunteers from Europe in order to provide genome sequencing throughout the course of this epidemic. And this was in Guinea. Now, the important thing if you're doing genomic epidemiology is that you need two things to make this work. You need to be able to sequence the samples quickly, because if you're waiting months to collect up samples to sequence in a big batch, then the information that's contained in those genomes is not so useful, because those transmission chains have moved on and things have moved on, and they're not so actionable. So the idea here is that we have a very rapid time to answer. We can sequence genomes within a day or two of a sample being collected and analyze that information. And the second important thing is that you have really good coverage of the outbreak. There's no point just sampling a particular village or a particular town, and then you don't get a feel for what's going on nationally and internationally. So when we got set up by about May 2015, we were able to sequence about half of cases. And in so doing that, because pathogens evolve quickly, and Ebola virus is a single-stranded RNA virus, it evolves very quickly, rate of about 2 mutations per month, we generate a large amount of genetic diversity when we look over the period of a couple of years of outbreak. And what you can see from this phylogenetic tree is you have a large amount of structure in that tree, and that structure, this is color-coded by country, is very much associated with individual countries, but you can see examples where you have mixing of, here, purple is Sierra Leone and green is Guinea, down here as well. It's strongly indicative of cross-border transmissions. And we can go into a much finer grained detail and provide information to epidemiologists in real time, showing when new cases arrive in a place, where they came from, and whether they're part of an existing outbreak or a part of an existing chain of transmission, or part of an unmeasured chain of transmission. So I wanted to get to this video, which was done by Gytis Dudas, working with Andrew Rambo and Philippe Lemay, where they reconstructed the outbreak using 1,600 genomes from the West African Ebola epidemic, and using time information, using place information, and case count information, generated this animation. Green is Guinea, blue is Sierra Leone, red is Liberia. That's based entirely on the This is generated from that phylogeny, from the phylogeny and the place, and trying to model transmissions between place according to movements. So large movements here in Guinea, even towards the tail end of the outbreak here, very large movements to far-flung corners of Guinea. So kind of, it looks a little bit like sort of, intercontinental ballistic missiles, and that's because the model they use. But the important thing here is firstly, the kind of pattern of spread, which is that you see a large number of transmissions over long distances, and those transmissions either set up a new outbreak in a local area, or they kind of fail to catch light and they peter out. And that seems to be the model, or certainly at least in Ebola in West Africa. And this is the model that we would like to see for outbreaks generally, globally. We think about things like AMR, etc. This is how we would like to be able to look at data. How much time have I got? Who's on the clock here? Yeah, we're quite. Okay, good. Okay, no worries. That's fine. That's good. Okay. So in terms of source, so Ebola looked like it was winding down by the end of 2015, and in fact, Guinea had been free of cases for a good several months until spring of 2016, when a new cluster of cases occurred, and we were able to respond to this new cluster very, very quickly with the real-time lab. And what we were able to show is that these new cases appeared in a part of the tree that we hadn't actually seen for quite some time. Most of the cases we've been looking at have been in these big lineages, GM1 and SL3, but these new cases appeared to cluster with much older cases. And we look at that as a more fine-grained phylogeny. What we can see is we have a bunch of cases that are all either indistinguishable or different from one SNP in March, April 2016. And if we go to the most recent ancestor, that's a survivor case. And if we go back one more, that's a case from exactly the same region. So we can immediately say, actually, this is a case that comes from, it looks likely to be linked to cases that were in that region, but we have this, something weird's going on here, because we either have a massive sampling gap, and there's been a load of cases that we've missed, or something weird's going on. What we can say is it's not a new outbreak, it's not a new spillover. These cases are very clearly within the ongoing outbreak and the genetic diversity of that outbreak. And so the weird thing that was going on here is in fact survivor transmission. So we were able to report back that these cases seem to relate to this person and relate to this area. And the epidemiologist said, yes, actually there is a link between this person and these cases. There is a very close link. On testing of the survivor's seminal fluid, it was found that they were strongly positive for Ebola. And so this is an example of Ebola persisting in the seminal fluid for 500 days and then resulting in a transmission, something that no one thought was possible. Usually Ebola would be cleared within 30 to 60 days at a kind of maximum. And we could show with the genetic data this was almost certainly the reason, and combined with the epi data, we could be pretty confident. And if you're eagle-eyed, you'll realize that this branch length is too short. I told you that Ebola is mutating about 2 SNPs a month, but this is 500 days. And in fact, what we see when we look at the root to tip divergence, this new case is way off the plot of where we would expect the virus to sit. And this seems to be now a well-established fact, which is that survivors who have Ebola seem to, their Ebola virus seems to evolve during persistence at a mutation rate, evolution rate of about 1/5 or 1/6 that we see in human-to-human transmission, which is an interesting finding. All right, so just to finish up really, we use this technique, nanopore sequencing, to look at Zika as well, and rather than using fixed labs, we moved to a kind of portable lab setup. I mean, of course, none of this is as cool as doing it on the ISS, I realize, but we did think that it would be a technique that's amenable to portable sequencing, and of course it was. And the point about this slide in Zika is just simply to show that after we'd collected a whole bunch of Zika samples and sequenced them, we were able to reconstruct a phylogenetic tree. And in so doing, we were able to show that at the point that Zika was detected and that association with microcephaly was made, sometimes around April 2015, the first clinical case, when we actually draw the phylogenetic reconstruction, we see a vast amount of genetic diversity and Zika affecting, we infer, affecting a large number of different regions of South America within that timescale. And we can date back the introduction of that Zika virus all the way to the start of 2014 or earlier, hopping across the Pacific Islands into Northeast Brazil and then across the Americas, sweeping across the Americas. And what we're showing here is that this is a common pattern, different regions. It takes a long time for the first clinical case to be detected, but actually the introduction into that area happened a good year before in each case. So to finish up, our big idea really is that this mobile sequencing technique is going to be important for sequencing in the field and maybe for sequencing in space. We've identified a whole bunch of putative difficulties in doing this that we're attempting to address in my lab and other labs as well. And we've started to build a prototypical kind of clinical microbiology lab in a suitcase. And this is just really to show the progress that we've made. This is working with Ian Goodfellow in Cambridge. This was his first version of a lab in a suitcase, where he put an iron torrent on the back of a flatbed truck and took to Sierra Leone. It was then compressed down into what I call lab in 8 suitcases for an outbreak that we investigated in 2017. And our current version is this size. And the difference between this and Josh Quick's luggage is that this is everything we need to process raw samples safely and without the need to take any extra equipment, enough for 100 samples. Okay. So last slide, I think transmission studies are important. They're really, really useful in clinical microbiology. They're really useful for looking at outbreaks, either national, international, or at a local level. And phylogenetic studies kind of get over the problem of statistical studies in space because they're extremely powerful for source attribution, certainly if you've got a database to relate to. And these strains that we have can adapt very quickly, and the type of strain that an astronaut might be infected with have implications for treatment source. It should be possible to do these kind of transmission studies directly from clinical samples in the future. So to finish, thanks to all these people that did the Pseudomonas work and all these people that did the outbreak surveillance work. And thank you, sorry, going over time.